Hello.
Hi.
Hi, how are you?
You also found how am I? I'm good. We had a winter storm here, and the dogs refused to go out of the house.
Oh.
Had to bribe them to get them out of the house.
Yeah.
It's still blowing.
Oh, wow. That's horrible. Ah, spring.
Yeah, it's spring, except it looks like winter.
So, how are you proceeding on your stuff?
Well, I found a paper that showed me how the formulas worked for at least a three bar tensegrity object, and I've made one, and then it gave me all of the formulas, but there's an iterative formula included in that, and I need to figure out how to put the iterative formula in console. The rest, I know how to put the rest in,
Yeah.
exactly where to put the rest of the formulas, but the iterative formula is like, I know console does iteration, that's sort of what it's based on, but I don't know where to put this formula. So Um, hopefully somebody will contact me from console and tell me how to put the formula in the physics section of
Yeah.
the simulation.
They don't have a user forum?
Yeah.
Didn't get a good answer.
It's not like MATLAB. MATLAB
Okay.
has like it's perfect. MATLAB, you get an error, you copy paste it, and you've
Yeah.
got actually they give you the workaround programming. But in console is like, oh well, maybe you do this and maybe you do that, and it depends on this. Is like, okay then. So,
Yeah.
anyways,
Well,
having trouble with console here. If anybody knows how to use it,
yeah. I don't think anyone does, but in our group, but yeah, I don't know. have a passing familiarity with it, but it's not something that I've worked with in the recent past.
okay.
So
Well,
it's
they've got a new version out. It's got all kinds of interesting bells included, but um, yeah,
yeah.
you can make an app using it. Show your work.
Uh, I've used MATLAB and SolidWorks, but not Console. Console, I think, is more advanced because yeah, I think FEM models and those kind of things. It's like
Okay.
even more advanced rendition.
Well, this is like I said. I have the formulas. I just need to figure out how to put them in the physics of the model.
Okay,
So
okay,
I I'll just keep trying.
okay.
I don't know.
Yeah.
Anyway, that's what's up with me. That's all
Okay.
that's up with
Yeah.
me.
Yeah. That's fine. Well, that's good. I mean, you're sometimes you make progress. Sometimes you get like caught up on things, and like that's yeah. So, Karan, how are you?
Oh, is he there? Oh, I'll shut off my
Hello.
Hello.
noise.
Yeah.
Yeah. Hi. 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 Yeah. Yeah. So yeah. I'm doing good, Bradley. Still, the axis rotation thing. That part is still like I'm currently working on that only right now, because the thing is, if I the current model rendition that is coming, it's the outlines, if they're not aligned under that particular axis of rotation, they will some will protrude in a weird manner, like that was visible in that model, like the like for one particular side it's showing the perfect embryo projected image, but for the area between that area, it's not coming properly. So I'm still working on aligning that properly first. So the contour that is there.
Yeah,
Uh,
I'm going to. Oh, sorry. Have you tried the images from my other paper at all? The
uh,
peppercorn and the flower. That
okay, no, I don't have those images.
Oh, okay. Well, I'll make. Okay. Well, and I didn't get my new images either. The power was turning on and off here due to a winter storm, so I gave up. I didn't want to ruin my computer, so that hasn't happened yet. It will happen this week, and I will get you those images.
Looking forward.
Yeah.
Okay, so that you're not fighting with odd angles from the flipping microscope, because they change, because the wheel didn't always engage, and if you made it too close, if you put too much pressure on it, then it wore out. Like it was the whole thing needed to be more professionally put together, I guess you'd call it, for it to work properly. It was a concept only. So this one, because it stands still, with the fixed angles, it should be easier for you to work with.
Okay.
Yeah.
Because the angle of rotation is already predefined. So.
Yes, the angles are 90 degrees on the top and 90 degrees on the bottom, and then the top and bottom one. So it's the angles are, well, they won't be precisely like that, because you have to change them so that you can actually zoom in on the object, so that, but they stay the same, which is useful. Once you've got the angle, it's not going to change. Because it's stationary. That's why I built this. I know I have had enough of moving parts.
Okay. Yeah, that'll definitely. Because the angles are predefined, then so it's just we know the projected outline from each angle, right? So then it's just mapping the contours properly. Then that won't be too much information. Because the axis is also fixed, it's not stationary. So, and even an interesting thing that Richard Gordon had pointed out was that they're not rigid structures, So if they're rotating very fiercely, maybe their actual size and the size that has been captured on the microscope, that could also change, because the centrifugal force is kind of.
Well, it used to dive. It used to take a dive, and then it wouldn't be in focus. I eventually put a little bit of plastic in a tube in, so that it wouldn't take as deep a dive. So it would stay in focus better. So, it was somewhat difficult to work with. You got. I could use a 24 megapixel camera to take the images, that's all. So you had better resolution images with it. Because I got a microscope tube for the lenses from a company in Europe, and they guaranteed that the higher resolution images. And I'm hoping with the 10 images at 2 megapixels that maybe it'll compensate for that. Just no digital zoom on it.
Even with the current data set, at least a good approximate model, like it won't be that much difficult now, when I'm trying to approximate the axis of rotation. So the only part is meshing all those projected images together, in a singular equilateral image. So that would greatly benefit with more samples, but the model will remain the same. It's just like when we're seeing an axolotl image, right? The pixels on the outer edge, closer to the outer edge, right? They're not like there's not much information that can be taken out from that. Like the one that is in the center, which is in the focus area, that is clear cut because that's what we see. But towards the edges, those pixels, they need a lot more augmentation techniques to work around with, so that I can get the proper thing out. But the model is it's on its way. I think maybe by next week I'll have the proper model. I just have to tweak it with a few more things.
All right. Well, that's great because we'll just try to put all this together.
Yeah.
And I have a friend who has a granddaughter who has salamanders. I said, "Oh, would they like a friend?" So we'll see if I can get some
Okay.
names. Yeah. Anyway,
Okay. Okay.
yeah. Well, I'll see.
Okay.
Well, that's good.
Yeah.
Thanks for that
It might be
update.
fun to work with
Yeah.
salamander eggs.
Yeah, it would be good if I could get the actual thing I designed this for. Yeah.
Okay.
Or maybe I'll find an antag and it'll do something. I don't know.
Okay.
Someone suggested spider eggs to me. So.
Okay. I have no clue about spiders.
No, well, they're fun to watch have, but
Okay.
I like, yeah, we'll see.
Yeah.
I'll try to find the salamanders first.
Okay.
Maybe
Yeah, because it might be
it's
closer in.
seashells as well. So
Oh,
I'll get some images of peppercorns and seashells
Okay.
for you.
yeah.
Yeah. That definitely helps.
Okay.
Yeah.
As long as the storm doesn't totally wipe out my computer, I'll be good. Yeah, I have an uninterruptible power supply on my computer, but even then I was just worried about spikes because I've had it take out my computer before. Actually, this one, it crashed my computer. My D drive just was gone, and anything I stored on it.
Okay.
Yeah, so we had a storm here, and it was snowed, and there was an ice storm in the middle of it, and there was thunder and lightning. Everything.
Yeah. Wow.
No, my dogs refused to go out of the house. Anyways, I hope the weather's done being stupid
Yeah.
for a while.
Hopefully.
Yeah.
We have
Anyway, that's
we have a white spring here.
Yeah.
a white spring. Yeah.
So, thank you for that update, Karan. Jiahang, do you have anything to update us on? Sort of things you've been doing?
Oh, hi. Hi. Yeah, I have some, maybe there are some problems with my network connection. Yes, maybe you cannot hear me clearly, but if you
I can hear you, yeah.
if you feel that you cannot hear me, you can just type, and I will know that. And I do remember that I proposed a research idea about combining the sequence-to-sequence and graph generation, and we can employ this idea on the cell movements prediction. Yes, you've mentioned this topic in the proposal of graph neural network projects, but we didn't focus on that. It's because I told you that there are a few works of deep learning using deep learning techniques to solve this kind of problems. So I think maybe we can look at this problem and maybe we can try something, I think.
Yeah, that would be great. I know I saw that you submitted a project proposal, so I'm going to read through it and give you some feedback on it in the context of this. I know that we mentioned this in the Slack as well, the seek-to-seek solution. So we'll talk about that. Yeah, that sounds good.
Yes, yeah. I mean, maybe there is some time before the project started to coding. I mean, actually, this research idea is separated from the project. So I think if we are interested in both of the project and the research idea, maybe we can work on it concurrently. Yes.
Yeah.
Yeah. And just like what I said, there are a few works on that, and actually there are a lot of scholars in the field of graph neural network are focusing on the graph generation. Yes, it is actually a trendy things.
Yeah.
So I think maybe we can try something like that. Yes, and actually I've surveyed a lot of papers, and I found that there are few works on combining the graph neural network and like the dynamic or the developmental analysis of biological things. Yes.
Okay.
So I think there are lots of we can try. Yes, that's what I mean. Yeah.
Yeah, that sounds great.
Yeah.
Could you put together a list of things that you've read? I know that you said you've surveyed some of the literature.
Yes, I can put those things on Slack. I mean, after the meeting.
Okay. Yeah, I mean, yeah. I don't know if you can like make a list and put it in a list, or you can put the papers in as well. But it's usually like if we have a list, I can get the papers. It's like you have a bunch of papers in Slack, but that's okay. It's up to you.
Okay. Yes, I will do that. I can try to. I mean, I can try to have links of these papers in a like Markdown. Yes, and I will share it in the Slack or something else. Yes.
Okay. Yeah, it sounds good. Yeah, I'm glad to hear that you're making some progress on that to find things and make connections between things. So.
Okay.
Yeah.
Yeah.
All right. Well, thanks for the update. So I was able to get the deadline for the GSOC projects was Tuesday, and I got your projects. I got all the applications that people sent in, and we'll be making a decision on that in the next week or so. Which ones get funded? Which one? This is actually up to INCF for them to make a determination as to what gets funded and what doesn't. So, but we'll release those results in a couple weeks, and then we can still work on projects if people want to work on them, whether they get funded or not. I think we've been doing a lot of good work here, and there are other opportunities for funding, whether people get GSOC projects or not. So if we get a GSOC project, that's great. We get like a summer where you get paid for it. If not, we can always find other opportunities to get funding or to, and then there are always publications that this will lead to. There are all sorts of opportunities. We've had, like I said, the DevoLearn project was the culmination of a couple years worth of GSOC projects, and I want to add I want to have the graph neural network work in sort of incorporated into DevoLearn at some point. So we would be building off of that as like kind of a module. So people could, download DevoWorm or download DevoLearn and then have like another set of libraries that they could use for the graph neural networks. And then there are some other things that we have, some other algorithms that we have that we can maybe add in later as well for other organisms. I mean, we've actually in the GitHub organization for DevoLearn, we have those sort of already in there, but they're not like in the same release. If one were to download the release from say PyPy, it just includes the stuff for DevoLearn, but there are other libraries. So, that's something we're working towards. So, I think I hope to be kind of a resource where people can download like a pre-trained model for deep learning, have some other libraries that also have some deep learning algorithms, maybe for some other broader set of organisms, and then have the graph neural network so that they can derive graph structures for their data. I mean, that's I think that differentiates that line of work from a lot of the other stuff that's been going on, because a lot of people have been sort of embracing this area of deep learning and microscopy analysis, and there are a lot of people doing things with it, but it's largely like segmentation, and I don't know if there aren't a lot of pre-trained models. There aren't really too many graph neural network models, so that's why I want to make sure that they eventually become part of a, sort of a family of things that you can download and use. But that's the long term vision for that. And then for the digital microsphere project, that would be nice to have as well. Maybe something that like a web interface that people can go to or download. And it wouldn't be really in the same family of approaches, but it's something that we could include in the DevoZoo. And I think you've gotten the impression that in like in development there's so much diversity of what's going on. Like there are so many different modes of development and organisms that we need to have a range of approaches that people can use. So
yeah. Okay, Bradley.
Yeah.
Next question. Harry Krishna and I talked on Saturday,
okay.
and he's run into a problem superimposing images on a sphere from Susan's data.
Okay.
Okay. And I suggested a technique he wasn't familiar with, which is simulated annealing.
Oh yeah.
Okay. The idea would be to use the spherical coordinates and move in the space of spherical coordinates and have some matching criterion. And then if it hit local minima, simulated annealing might get over them. Okay.
Yeah.
Now I was wondering if there's something better.
Oh. Well, I mean, they're like in terms of optimization, there are other optimization techniques. I mean, there are a lot of different types of optimization techniques.
Yeah.
But I don't know if there's something that's like simulated annealing but better. So it's like you can use that basic approach. And I don't know like the state of the art for simulated annealing. I'm sure people have worked on this a bit more in the recent past, but maybe not. You could throw like a regular set of optimization algorithms at it.
Well, let me ask a question. What algorithms are used for image montaging?
I'm not sure.
When you try to sew together a set of images to make a big panoramic image.
Yeah. My method is that kind of using these ideas only. It's like let's say there are 8 images per sample of an axolotl embryo, right? So we'll have 8 perspective images of the axolotl embryo. Then what we do is like you've seen the projections of Earth on a globe and a map. So we do a projection transformation. We get an equilateral rectangular perception. It's like it uses coordinate transformation to a cylindrical projection area. Like from a globe, we'll project it onto a cylinder. Then from that cylinder, we'll just lay it out linearly so that we get a very big panoramic, image.
Oh,
The problem that I'm trying to solve currently with the current axolotl embryo data set is, since there are 8 perspective images, I can convert all of them to those equilateral rectangular projections, rectangular projections of that spherical thing on a plane. The tricky part right now is since the side pixels are also kind of distorted, because I think resolution plus the angle of rotation aspect also. So mapping them correctly with each other, stitching all these 8 images together is the main problem that I'll try to cover right now. It's one of the otherwise working with more images, kind of improves this model. So let's say if there were 12 images, so we'll have more, overlapping points. The more overlapping points we have, the easier it is to use, OpenCV, like computer vision algorithms that I'm currently using, to stitch these images together. Because they have more data to work with. So, they give a better approximation.
Could I ask you a favor then? contact Hari Krishna and
Yeah.
send him your ideas, because he's been stumped, and all I could think of was the simulated annealing approach,
Okay.
Yeah.
which was new to him.
Okay.
And by the way, I guess the cylindrical projection would be analogous to a Mercator projection.
Yeah.
Yeah.
Otherwise, the different. There's another orthographic way of doing it.
Yeah.
Orthographic projections are there, which I think the closest approach would be similar, like the cylindrical projection to an equate rectangular projection. Then getting
Mercator is to a plane, but you're saying it's similar to a Mercator projection, but to a cylinder.
Yeah.
Okay.
Yeah.
Okay. Anyway, I'd appreciate it if you contact him and throw some ideas at him.
Yeah, I'll definitely do that.
Okay.
I'll contact him on Slack.
Yeah. Yeah, there's this whole emerging area of 360-degree videos where they take video from different locations and then they put it together and you can walk through a scene and rotate around it and you get a view, and it's pretty seamless in terms of the perspective. So you're moving around a scene.
Yeah. Whatever happened to the field of gigapixel imaging, which is analogous to that?
I'm not sure.
Okay.
Gigapixel, you take lots of images of a scene and stitch them together, but you retain the original resolution so that you can zoom in and see things close up.
Oh.
But the image will be gigapixels in size.
Okay.
Yeah.
Okay. That was a fad about 10 years ago.
Okay.
Yeah, I don't know.
I think they must be using it in current AR technologies. It's kind of all Meta is there, Metaverse, and they're trying to augment both of these aspects, especially with 3D reconstruction techniques. Maybe they'll have something in that.
Yeah, they would do things like, I don't know, take a high resolution microscope and automate its controls so that it could take a large object, but put together many small fields into a huge picture. So it retained the full resolution of the high resolution pictures, but it was a huge picture, which you could then examine at any resolution you wanted.
Okay.
What about photogrammetry?
I don't know. I mean, I don't know anything about it, to be honest. What can you tell us?
It's just you can take, it's like you have two eyes, so that will give you depth of field.
Yeah.
So it's similar to that. You have to take several pictures and then you have depth of field.
I've kind of compiled, a very for this for 3D reconstruction only. I was kind of going through a lot of different methods. So should I share the link for that?
Yeah.
Copy link. This has the majority of the reconstruction techniques that are used, right? There's one called structure from motion, in which you have a very random array of perspectives that you've taken, from a camera, and the software tries to make a point cloud, like a 3D point cloud of the things that it has seen. Similarly, Susan was mentioning, two cameras to measure the depth aspect of it. That is for MVS, multiview stereo vision.
I had a student years ago who we wrote a paper on doing that from one view.
Oh.
Okay.
I don't know how he's getting spatial information.
I can tell you how. What you do is you do a computed tomography reconstruction in 3 dimensions. Okay. And you do it with very limited data, which means that it's not a very good reconstruction. However, if you take a particular view along one of the directions that was used for the computed tomography, and then add 3 degrees, subtract 3 degrees, and take those views of your reconstructed data, you make a stereo pair. And then you can see depth.
Oh
yeah.
This is a nice take on the.
Can you get a reference for that? That sounds very.
His name started with J.
Because I've seen some ML models, doing something like this, but even then, they're trained on images or they already know what the model is supposed to look like. So they kind of make closer approximations.
Oh, no, in this case, you don't know.
Okay, you don't know, okay.
But it was kind of nice. It actually produced a decent stereo pair if you looked in the right direction.
Yeah.
Okay. Send me a reminder, I'll dig it out. I just remember the student's surname started with J.
There we go.
Okay.
Yeah, you want me to share this, Karan?
Yeah, this is the list of, various 3D reconstruction techniques. I've kind of most of them are currently used for projects like either drone mapping is there, like current SFM techniques are used for drone mapping, open source software, drones which take a lot of images and then, construct a 3D model of whatever has covered, area mapping drones. Similarly, so this is kind of like a list of different ways, people have approached reconstructing 3D models. So, while I was going through my axolotl embryo, like I was looking for ways to reconstruct the model, I kind of got this list of methods. So structure for motion is there. Structure for motion is basically a camera that is moving around and has different perspective images, that are unsorted. Otherwise, we usually have a sorted array of a particular angle and a particular plane from which, the camera is taking pictures. SFM otherwise uses an unsorted method for that. Similarly, down below we have, I think, MVS is there, and SLAM. SLAM is also pretty popular. SLAM is, I think, the base for current self-driving cars. They try to visualize using SLAM techniques, if I'm not wrong.
Yeah.
So this is also pretty active, this area of research. But since, my data set is kind of very different because they rely a lot on, LiDAR sensors, additional ways, to get any sort of distance or depth information, all those things.
Yeah. So does SLAM require like dynamic data, or can it be used like on something that's static?
It's like I've seen SLAM techniques being used for again, the data is static in the sense that you'll have a scene image of different angles from which you've taken the picture.
Yeah.
But it's kind of the stitching together part and constructing the depth information part. That is kind of unique. That has been approached in this method.
Okay.
Okay, I put the reference in the chat.
Yeah. Okay.
So this is display of 30 anisotropic images from limited view computed tomograms. So that should be Jaman as the first author. Yeah. So then there's multi-view stereo vision, which is what can you tell us a little bit about that?
Yeah, multi-view stereo vision is it's like it uses visual audiometry techniques only in the sense that it has two cameras that sense the for taking simultaneous images, and then it kind of creates a depth field of some sort, But they use more like for SLAM you have continuous data, time continuous data. For multi-view stereo vision, you have instead of taking just one perspective of that particular camera, you have like 2-3 perspective views of that same scene, that you're trying to model.
Right.
So it has slightly lesser data and it makes slightly more approximations as to what the model will look like. So point cloud computation, making the surface mesh, based on those point clouds. Sometimes a lot of outliers, like if I think I was trying a GitHub project based on this. So they were kind of mentioning like unless the camera has been calibrated properly, because camera also has kind of like 2-3 properties, because of which it kind of distorts the image in a positive way or the negative way. Where the image will either go inside the plane or come outside the plane. So those kind of details, if they're left out, it kind of tends to create more inaccurate models.
Right. Right.
Yeah.
So, that's good. That's a very good list. Thank you for putting that together. And the link is in the chat if you want to check it out, or I can send it along. Yeah. That's great. Thank you. All right. So that's
yeah, I'll definitely do that. Email address, yeah.
Okay. All right. I was going to point out something. This is going to the graph neural network stuff again. So there's a target conference we might look at. This is something that was announced recently. They want to start a Learning on Graphs conference. And this is something that, the graph neural networks area, they've kind of been sort of under the guise of a lot of the machine learning conferences. So they've had workshops at NeurIPS, for example, that have been very well attended. And so now they're going to start a virtual conference this year. They're trying to organize it and get everything going. So this is something that, they're trying to kickstart this area of research in terms of its own conference. So there are a lot of different, there have been specialized workshops in the past having to do with graph ML, like in industry, learning on knowledge graphs, molecule discovery, graph learning benchmarks. And so there are links to all these in this blog post here, but they don't really have any way to unify the subfields. So people would attend these workshops, and the conversations would be limited to those areas. What they want to do is create a sort of a large conference that covers all of these areas. So they're soliciting papers. They want to use open review for the review process, and then hopefully they get a pretty good set of submissions. So the deadline for this, I guess, is tentatively in September. So this is a couple months off. I mean, maybe we can put together a paper on this based on doing some work over the summer. I don't know. This looks like you have to submit it by September, and then you have a revision period here in October, and then the final decisions are released in November. I like that they allow you to revise the papers before they give you the final decision, because a lot of conferences, they don't let you do that. It's just like, here's the paper, is it acceptable or not? Which is different than journals, because journals usually allow you to revise a little bit. But anyways, this is so they don't have, I don't think they have, the submissions aren't open. I'm not really sure if they have the sort of the details on it, but I think the idea is that you're going to have a couple of different tracks. You might have an extended abstract track here for smaller submissions, and a proceedings track, which are longer papers of up to 9 pages. So this is something to think about as we're working on this graph neural network stuff. Thinking about how we want to, put together some sort of publication on it, or some sort of report on it. So, and then, this would be, I think, a really good target. It looks like there are all sorts of different approaches here, from generative models to knowledge graphs to expressive graph neural networks. So, I mean, basically, it's just about anything you can think of. So that would be great if we could target that. And again, this is something that if we don't get accepted to this, we have the paper, so it's not like it's going to be a waste of time. It's just a matter of getting it, getting something off the ground and getting something tangible out there, and see where it goes. So that's good. So I know we've talked about cell tracking and lineage construction, and this kind of goes back to some of the stuff that we've talked about with graph neural networks, but it applies to other things. And last week I covered the data that we have. So we have a lot of data that's available for embryos. Some of it's like the axolotl data, which is, it's kind of raw images where nothing's being tracked, to the C. elegans data, which we use, which has this cell tracking aspect to it. And they have cell tracking for all sorts of organisms. For embryos, they also have cell tracking or behavioral tracking, actually. So they use similar techniques for tracking behavior. In OpenWorm, we have a version of this for worms. It's called the Terpsy Tracker, and they take images of worms in a culture dish, and they track each worm. They use some marker on the worm, and they look at its movement, and they can track the movement around its environment. And so you can do this for other organisms as well. You can take high quality video of their enclosure where they're behaving in, and you can track their behavior and follow different trajectories. And this is all sort of enabled by deep learning technology, and in the case of embryos, also a lot of the fluorescence microscopy that we use for looking at gene expression, but also looking for other markers of different structures in the cell.
Bradley?
Yeah.
I'd say something. Hare Krishna's, if he succeeds with the montaging, then one obvious step is to segment all the cells that are on the surface and track them.
Yeah.
Okay, so we'd be tracking over a three-dimensional, spherical surface.
Yeah, that would be good.
Okay, then we could track things like cell divisions, cell size. We don't need any marker other than the cell boundaries.
Right.
Okay, which are presumably what will be used for the montaging anyway.
Yeah.
Okay, and then we can also see if some cells disappear, if any die, etc. And also when differentiation waves occur, which cells participate in them and what do they do and when.
Right.
Okay, so there's a whole bunch of questions we can ask once that succeeds.
Yeah, that would be great. All right. So let's see. So this paper actually is one example of something that people are doing with this technology. The C. elegans work, people have published papers on this. They also published papers in zebrafish, and this is for embryos, so you can go look at those. I think Philip Keller is a person who does this in zebrafish. So this paper is by a number of people here from Homogenelia. So this is a lab run by the Howard Hughes Medical Institute in Ashburn, Virginia, which is near D.C., Washington, D.C. And so this is automated reconstruction of whole embryo cell lineages by learning from sparse annotations. So in this paper, they present a method for automated nucleus identification and tracking in time-lapse microscopy recordings of entire developing embryos. So this is the sort of stuff that we see with, they're using, in this case, they're using stacks of images. So they're going from the dorsal to the ventral side of the embryo, going through a different different focal planes and taking high-resolution images. And then that allows them to get like an image of every cell because you have cells in different positions in that axis, cells across the anterior-posterior axis, which is front to back, and then to the sides. And so you can get every cell in the embryo that way. But then you also have things in time. So that stack is just one time point, and then you go in time, and you can actually look at the entire embryo. So their method here combines deep learning and global optimization to enable complete lineage reconstruction from sparse point annotations, and uses parallelization to process multi-terabyte light sheet recordings. So they're using this, what they call light sheet microscopy, which is a form of microscopy. It's high resolution. It allows you to see cell boundaries pretty well at that resolution. And more critically, it allows the algorithm to pick up on those boundaries and do the segmentation pretty pretty efficiently. We've used a lot of light sheet microscopy for our training data sets for DevoWorm, for example, because it's very easy to see things, or for the algorithm to see things in there. So in this case, they're demonstrating their technique on 3 common model organisms: mouse, zebrafish, and Drosophila. So Drosophila is the fruit fly, zebrafish is the zebrafish model, and mouse, of course, these are all common model organisms. On the most difficult data set, mouse, which has a different mode its mode of development is more like human than the other two. You get a lot of cells that you can't really track the cell lineage. You can track it using like a barcode or something like that, which is a technology that allows you to trace cell divisions. But the cells really, depending on where they are in the organism, they will, adopt their fate. So it's not deterministic like in C. elegans. So you can't really say that like this developmental cell will become this adult cell. It really is like you get a bunch of precursor cells, and then they can become specific cells in the organism with a specific position. And then they have these 3 different modes of development, different model organisms. So they're actually testing this out, trying to test for its robustness here.
Bradley, got a question about this work.
Yeah.
Are they able to mark the cells in any way in vivo so that they can tell what kind of cell it is at the time?
We'll probably get into that in a little bit, but
Okay.
yeah, I have to get in when I get into the methods.
Because if they can do that, then we can go from lineage trees to differentiation trees.
Yeah. So, on the most difficult data set, mouse, our method correctly reconstructs 75.8% of cell lineages spanning 1 hour, compared to 31.8% for the previous state of the art. So that they've been building upon the state of the art, thus enabling biologists to determine when and where cell fate decisions are made. So this is the main part here where they talk about some of the manual tracing lineages. So you can trace lineages manually using different techniques. Mammoth and Mastodon are the 2 competing methods. These methods are arduous, and for complex developing organisms, it is only feasible to annotate a small percentage of all tracks. So they have these different tracks that they're analyzing that are created in the program, and then they analyze it, making automatic cell tracking necessary for holistic analysis. So they've developed a number of cell tracking algorithms, while hand-engineered features are sufficient for cell detection and tracking. And so this Bao 2006 paper is C. elegans. This is one of the papers where a lot of the C. elegans cell tracking data come from. And C. elegans, of course, has that mode of development where we can do, where it's not as challenging to do cell detection and tracking. So, but they're talking about organisms where you don't know the lineage ahead of time. You have to actually map it out, and then you have to track the cells. So it's a little bit harder to do. Deep learning has been shown to improve cell detection, segmentation, on a wide variety of data sets. So so additionally, it has been shown that tracking methods that take into account global spatiotemporal context perform better, especially for data sets with more movement between time frames. So as the cells are moving around and going in different locations, cell migration is actually much more diverse than we see in C. elegans. This helps overcome that. So let's see. You can also do tracking by graph optimization over a large spatiotemporal context. This allows inclusion of biological knowledge about track length and cell cycle, improving track continuity, and even allowing recovery from noisy detection and segmentation. So why don't we get into the methods? So this is So to attain per voxel predictions for cell locations and movements, we use a UNet architecture with 4 resolution levels. To incorporate temporal as well as spatial context, we concurrently feed 7 3D frames centered on the target time point and use 4-dimensional convolutions until, due to valid convolutions, a time dimension is reduced to 1. So they're doing some downsampling. They use 12 initial feature maps. Then they upsample, so they do a lot of different things within the network. And so this is what these, okay, here it is. This is the data, the raw data. This is 3 dimensions in times. This is 4 dimensions. These are the reconstructions here without anything on them. Then there's a sparse point annotation where they superimpose the first frame over the first frame of the raw data. So they're reconstructing these graphs from the cell indicators and the movement vectors. So they're looking at the movement vectors of cells and the cell indicators, and they're looking at that in 3 dimensions. And then they build a candidate graph from that, which gives them information about like the movement and where the cell is in the embryo, and then they're able to reconstruct the lineage because they're able to like see like how the cells with this marker is moving, and they're able to do it that way. Trying to see if they have anything about like what kind of markers they're using. Candidate graph extraction, discrete optimization to find lineage trees. So, I think they're using like standard markers for the cells, and I don't know what they're doing this in vivo or what they're doing. I mean, obviously the embryo is, I don't know, it's under a microscope, but I don't know how they're doing this. I think they're just using standard markers in the cell, like fluorescent markers, to determine where the cell is, like the something in the nucleus. But I don't know exactly if they're using anything else to mark the cells themselves. A lot of this is like looking at the cell, how it, where it is in time. If you have, so for example, here you have a division event. As you have a single cell and then you have 2 cells, then you're tracking 1 and you're tracking 2. So it's, it's kind of messy, but you can see that there's like, 1 cell that divides into 2 cells. And there are ways to do this with like barcoding so that you can have like an identifier that's, unique to some cell that gets passed on to another cell. There are also in cases where you put a marker in a specific type of cell. When the cell divides, it passes down that marker. So you can tell like if a cell comes from another, comes from another cell, and thus from a lineage of dividing cells. And that's another way to do it. But then they take the movement vectors, which are the way the cells are migrating around, and they integrate those 2 into this sort of graph reconstruction. And then they have this ILP solution, which is an optimization technique. So they're actually able to trace out some of these lineages and things like that. So ILP is integer linear program. So this is the constrained optimization problem they're trying to solve. So there's a lot of computation in this, a lot more computation than cell biology. So this is largely just like taking microscopy data with some marker for the different parts of the cell we want to track, and then the tracking part is really kind of finding the position, putting it into a framework, and then reconstructing the lineages from that. So and then cell lineages can be used for a wide variety of analyses, and different kinds of errors can affect downstream results differently. This reduces performance of a tracking method to a single number that represents overall performance. You can't really do that because of all the different sources of error. So they actually have 5 types of tracking errors. So there are all sorts of different forms of error in cell tracking. So, aside from being a very computationally intensive method, it's also can introduce a lot of error in what you're actually looking at. So, there are ways to ground truth these reconstructions, but really it's a very tough problem to do. So this is why they're not able to get into like the 90% range of accuracy. But this method apparently has gotten, into the 70s. So considering the sort of how fraught it is with a lot of these error issues, it's actually pretty good. So that's good. I think that's just a little bit of a dive into that deep learning, some of the deep learning techniques that they're using, and something that you have to be one of these huge labs to really be able to innovate in. It's not something that's easy to implement, at least not today. We can do things with some things with deep learning, kind of like as a small group, but like reconstructing a whole embryo is really kind of a difficult proposition.
The person I'm working with, Dr. Sharif,
Yeah.
has he's done work with tensors, and he's been able to make sparse sets with the tensors that really work to eliminate a lot of redundant data.
Okay.
So there's I'm not sure of the details. I just know he makes lovely sparse sets so that computation is easy.
Yeah.
So that might have something to do with this. I'm not sure. Oh, sorry, my phone's going off.
Oh yeah. Well, I think yeah. So you always have this issue of sparsity in a lot of the biological annotation. So we have like annotation data. For C. elegans is pretty well documented, but for other organisms less so. And but furthermore, it's hard to know the process because there are a lot of things. There's a lot of signaling going on, and a lot of things that are dependent on where the cells are in space. And so that's where the sparsity comes in. It's just we don't know these different things. We can't even really measure them directly, but we still want to reconstruct the lineage to a reasonable degree. So, we're trying to get from 0 to something in terms of understanding these lineage trees. And we can do that, with all different types of data integration, but we can also do it with machine learning, with like optimization techniques, and saying this is the most likely path for this process. Okay, so yeah, Dick says I have another meeting at the end of the hour. Okay, so if you have to go, that's good. I mean, I can send you the papers and I can make sure that those get out. Thanks for attending.
Okay, I wanted to tell Dick that I ran across someone who's making small microscopes for dentistry that are actually optical coherence tomography. So maybe one could do optical coherence tomography from all sides of a ball, for instance.
Okay.
Yeah. Just wanted to say that.
Yeah. Okay. All right. So,
Good to hear that.
yeah. All right. So if you can stay on, we have one more thing I wanted to share with people. If not, you can go ahead and go. So this is something that I wanted to kind of go over. This is some of the things on the chemical brain hypothesis in early life. So I think Dick sent me this paper on systems biology and prebiotic networks. So we've talked about this topic a bit in the group. We've talked about like the origins of embryos and geologic time and how that organization might have happened. There are issues with like the great oxygenation events in Earth history. And that's one aspect of it. Another aspect is to go back even further and talk about what was happening at the origin of life. And so this is something that I'm going to kind of go through a couple papers on this to talk about this a little bit, about the systems biology. It's really interesting work. So this paper is on the early systems biology and prebiotic networks. The abstract reads, systems biology constitutes tools and approaches aimed at deciphering complex biological entities. It is assumed that such complexity arose gradually, beginning from a few relatively simple molecules at life's inception, and culminating with the emergence of composite multicellular organisms billions of years later. So there was an origin of life, and then there was a period where very simple life dominated and didn't really see very much complexity until maybe like a billion years later in Earth history. So it's really interesting how, that period, and if you've, like some of our previous sessions where I've talked about this, I make the point that this was largely due to some of these oxygenation events, where you start to, that the Earth in its very early history was very inhospitable to life, and it became more hospitable to life as time went on, and then some of the simple organisms were able to actually change the atmosphere so that it would enable more complex organisms that were based on oxygen and that sort of thing. So the main point in the present paper is that very early in the evolution of life, molecular ensembles with high complexity may have arisen, which are best described and analyzed by the tools of systems biology. They show that modeled prebiotic mutually catalytic pathways, and these are different types of pathways. I'm not going to get into what pathway analysis is, but these are biochemical pathways that enable living systems to sort of have metabolism, things like that. And they have network attributes similar to those of present-day living cells. So living cells really emerged during this time. You had these groups of, you had these small molecular structures that actually they weren't really cells, but they were like protocells, that they had these network attributes, these attributes of metabolic networks that are similar to present-day living cells. So that it suggests that there's a very long evolutionary history of this, and that it emerged early on. This includes network motifs and robustness attributes. So these are properties of networks that are often seen as optimized parts of a network. Robustness means that a network can overcome perturbations. Network motifs means that there are things that are grouped together that enable function in an organized manner. So these networks weren't just random networks, they had some structure to them. We point out that early networks are weighted, graded, meaning that they have some weight to them, so they have, they're not just like binary networks that turn on and off, but that using a cutoff formalism, one may probe their degree distribution, which is the sort of the structure of each node, how many connections each node has, and show that it approximates that of a random network. So these are actually, they look like random networks in the random network sense of network science. A question is then posed regarding the potential evolutionary mechanisms that may have led to the emergence of scale-free networks in modern cells. So scale-free networks are like, if you've ever heard of a small world network, where the networks have, they both have these sort of features of hierarchy, but they also have features of distributedness. So they're these kind of mixed network topologies. In this case, they're talking about things that approximate a random network. A random network is a network where there's no real hierarchical structure in the network. It doesn't mean that that they don't do anything or that they don't have structure. It's just that the structure isn't organized in any coherent way, as opposed to scale-free networks where there's a certain type of organization that leads to a certain type of behavior. So this paper goes through prebiotic molecular networks. They talk about some of the scenarios for early life, that life may have begun with a single molecular replicator, an RNA-like biopolymer that could make copies of itself. And it's further assumed that complex molecular networks came much later. So the networks that we see in modern cells, for example, came much later. So there's some gap in there in between these biopolymers and these very complex metabolic networks that we have today. And so this idea is that they're trying to model this lost period. And so they want to understand if the network came first or if other things came first. So it is further assumed that complex molecular networks came much later and were genetically instructed by the replicating polymers. The second set of scenarios asserts that early replicating entities must have constituted complex molecular networks right from the outset. The latter view claims that the emergence of single molecules, whose inner works allowed them to instruct the synthesis of their own copies, are extremely unlikely under prebiotic conditions. So it is claimed that the spontaneous accretion of specific mixtures or assemblies of simple organic molecules capable of self-reproduction is more probable. So this is where you had just kind of the spontaneous life that formed from spontaneous mixes of different things, and it just kind of happened that way. And so this is dependent on these assemblies having a certain set of network properties. So these are something that mimic present-day cells. So basically what they're saying here is that the metabolic networks that were in this early stage not only mimic what we see in modern-day networks in cells, in metabolic networks, but that this is something that's probably the way that this works. I mean, there's no other alternate way. It happened very early, this type of organization, and it just became a little bit more complex over time. So the network actually is something that you can actually see patterns of how life emerged, but also that those networks really, you can't organize this any other way. So they kind of go through some of these networks, and they kind of, this is very heavy in metabolic networks and analysis of those networks. So I don't really want to get into that. That sort of, I don't want to get into the technical details of that. But this kind of shows like prebiotic and contemporary biological networks. So I think they don't really have good. Oh, I guess this is the legend here. So this is a graded autocatalytic replication domain system here. So autocatalysis it's a very rich field, and they talk about this in complexity theory a lot. It's or in biochemistry even, and it's the idea that you have this catalytic cycle. You have to have certain things in place for a catalytic cycle to occur, and this is a autocatalytic network. So this means that this is a network that's wired to sort of perpetuate chemical reactions, and so this is what it looks like when it's sort of self-organized. So this is what they call the composome. And this is something that I'm not going to get into, but this is a word that they throw out. So I guess that means it's composing itself from different components. And this is something that they've been able to simulate and build together. And then this is the schematic illustration of this autocatalytic set. So Stuart Kaufman, who is someone who's worked on other types of networks, like NK networks, also proposed an autocatalytic set. And so this is something where you have a number of different sort of essential pieces that have to be in place for some of these catalytic reactions to occur. And so this is a minimal set of catalytic components that need to be in place. And so, this is a comparison with something you might see in a cell today. So this is a network. It looks much different than this composome here. This is a protein interaction map where you see different things. The structure is very different from this network. And likewise for this D, which is the metabolic network of E. coli, this is very different from this autocatalytic set as proposed by Kaufman. But A and B are actually the thing that's we see very early in life as the authors are proposing, whereas C and D are things that we're seeing in modern cells. And so that's... then the next paper is about the chemical brain hypothesis. So now we're moving towards the origin of nervous systems instead of the origin of cells. And this talks about what they call the chemical brain hypothesis. And so this paper, I'll just go over the abstract a little bit. In nervous systems, there are two main modes of transmission for the propagation of activities between cells. Synaptic transmission relies on close contact at chemical or electrical synapses. it is possible to wire complex neuronal networks, as they call them, which are networks of neurons instead of like computational structures, by both chemical and synaptic transmission. So your synaptic transmission have neurotransmitters. Your chemical transmission can be across the synaptic cleft, or it can be like out in the milieu where the cells live. Both types of networks are ubiquitous in nervous systems, leading to the question which of the two appeared first in evolution. This paper explores a scenario where chemically organized cellular networks appeared before synapses in evolution. So a lot of our computational neural networks depend on this synapse analogy. And what they're saying is that synapses may not have come first. It's actually these cellular networks may have come first, and synapses came later. So the evidence for this is the sort of peptidergic signaling that happens in most animals except for sponges. And you see this today. It's a very non-specific form of signaling in your brain, in neuronal networks. But it happens, and it's probably a precursor, or it's probably a remnant to that precursor network, that you have these peptides that are signaling each other, or signaling cells, and you don't have synapses yet, but you have chemical communication between cells. Small peptides are ideally suited to link up cells into chemical networks. They have unlimited diversity, high diffusivity, and high copy numbers derived from repetitive precursors. So this chemical signaling is diffusion limited, but chemical signaling is diffusion limited and becomes inefficient in larger bodies. So these networks had to be fairly small, or else they would become inefficient. So you can see as life maybe became more complex, these peptidergic networks became less viable, and so you started to have synapses emerge, and then that's how that sort of signaling actually became predominant in large brains, or in brains of any sort of size. To overcome this, peptidergic cells may have developed projections and formed synaptically connected networks, tiling body surfaces and displaying synchronized activity with pulsatile peptide release. Basically, this is something that is, this would be like a sort of a set of adaptations to the size limitations. So as the size of these networks got larger, they started to develop things that look more like a synaptic nervous system. And then synapses just made this all more efficient. And so you still have peptidergic signaling, but it's less predominant in most, complex organisms, we'll say. The advent of circulatory systems and neurohemal organs, which are basically blood flow and things that, you see in a lot of more complex organisms, further reduced the constraint imposed on chemical signaling by diffusion. This could have contributed to the explosive radiation of peptidergic signaling in stem bilaterians, which are these basal, our ancestors, the ancestors of C. elegans, and the ancestors of a lot of organisms that have this bilateral symmetry. So the stem bilaterians are a basal form of all, I think, a good number of animal species. Neurosecretory centers in extant nervous systems are still predominantly chemically wired and coexist with the synaptic brain. So neurosecretory centers are things that are like different places in the brain where you have the secretion of hormones, for example. And so this is actually, maybe this is the origin of hormonal signaling. But they kind of go through, so this is from this article, or this special issue on basal cognition that they had in Philosophical Transactions B. So this is a nice paper on this is from the origins of the brain, and it kind of goes through a lot of these things and how these peptidergic networks might work, and it's very different from the networks we just talked about, or like your typical neural network. So that's something worth looking at. And then there's this last paper on pre-metazone origin of neuropeptide signaling, which is from the same author with some other co-authors on it, and this kind of goes through this this idea of neuropeptidergic networks, where you see this in different types of organisms, that still have this type of nervous system. And they're using this as a model, the extant choanoflagellates that they're looking at. And they're using that as a model for evolutionary origins. So they're looking for two neuropeptide precursors with broad evolutionary conservation. So that's one way to look at, like, the origins of something, is to look at things that are highly conserved or the same across multiple species. And then you take that data and you hypothesize that there was a common ancestor. And because it's conserved, that means that you can predict the common ancestor without too much trouble. You can predict that they kept a lot of the same features, and so you can hypothesize a common ancestor with those features. So it's something they call deep homology, which is where you can go very far back into evolutionary time and assume that things are basically shared amongst maybe very divergent species. So, something that's shared between C. elegans and humans, and there are some things that are, in terms of genes, have what they call deep homology, which means they're shared. And so this is a kind of a tough area because it's hard to know exactly what has deep homology, and finding those things is very difficult. But this paper, they kind of go through this for this neuropeptidergic signaling. So anyways, that's all I wanted to talk about in terms of that. I kind of hope that gives you a sort of a nice idea about networks. And so Susan had a comment here. Early biology with regards to gene DNA development. Evolution was in the APS physics conference, A14 Monday morning lectures.
Yeah, I sent you that with the authors in the A14, like the Monday morning.
Yeah.
And I don't know if he was the second one or the last one, but they basically showed that certain gene sequences came out on top with regards to that kind of an evolution, and they were able to simulate that.
Yeah.
So it was interesting, and maybe if I think you should follow up on it. It's not really my area of expertise, but you and Dick have talked about it a lot, so I just thought that you might like it.
Well, yeah, thank you. I saw that. Actually, yeah, I've been working on that list of potential contributors. So you and Dick both sent me things, so thank you for that. And we got to get moving on that. I got to figure out how to start contacting people, and well, we'll get to that. So next week we'll be here again. We'll have if people want to present, they can bring something to the meeting, and or ping me on Slack, and we can talk about some things. And I hope everyone has a good week.
Yeah, I hope my console model decides to work.
Yeah. Okay. Take care.
Bye.
Bye.
Bye.